very brief overview of the nervous system. Remember that the nervous system initiates in the brain and connects to all of the neurons or nerves in your body. The brain, important for your health because it controls everything. The nervous system will vary depending on the organism. We're going to focus on humans. We have a brain, we have neurons, and we also have a spinal cord. Spinal cord is where a lot of our neurons will branch off from. So like damage to the spinal cord can cause, for example, to have issues like completely with your lower body, or if you have it up high, like in your neck, your entire body. The nerve cell is called the neuron. It gets a very specific name. They have a really cool shape. Neurons perform these 4 functions. From somewhere else they're going to receive information. Could be from the brain itself, could be from another nerve, a gland, or a muscle, or another group of cells. It's going to take that information and it's going to do one of 2 things. It's going to take that information and be like, that's not enough information for me to pass on a signal, so I'm going to stop this right here. Or if it gets enough information of a certain kind and it hits a certain level, it's going to pass on a stimulus to the next gland, muscle, group of cells, neuron. It's going to send that to the end, and then it's going to communicate with something else. The parts of the neuron. So let's take a look at this one, the light orange, or the lightest colored neuron up here. Look how cool the shape is. These can be feet long. I should say a meter long. So this could be a meter long. You have the cell body. The cell body, on its kind of branches out, they're called dendrites. The dendrites receive information from other places, integrate that into the cell body. The cell body will calculate out that signal. If it hits what we call threshold, it will send a signal on to its synaptic terminals. Terminal means ending, so I kind of like that. Synaptic terminal. Synapse means an area, there's actually a little tiny space between the dendrite and the next cell. So you have information that is shared in here. Via diffusion goes from high concentration here, high concentration in the synaptic area or the synapse, to low concentration here and gets absorbed via diffusion. Okay, so dendrites. Dendrites have a lot of surface area, branches, 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 branches. A lot of branching. We know branching means surface area. Increased amount of surface area so that it can receive a lot of information from a lot of different sources. You can see that they reach out in a lot of different directions. So this is pretty cool, and one of the things that always frustrates me about cells, when you study cells early on, you often think like, this is a cell. And it just acts by itself. It doesn't contact, right? Like you see pictures like this. But in reality, what we're talking about is this cell is really in contact with a lot of other cells around it. That's why I always hate that early learning is that you get stuck in your head that cells exist alone, but they're in contact with so many things, right? So change that in your brains. It's hard at this point, right? There's receptors, and we'll get to those in a minute. The protein receptors will take a signal receive it. If it binds, so anytime we're talking about receptor-wise, if we're talking about receptors on cells, that you might have a bunch of receptors floating around in that, what we call synapse area, between a neuron and another cell, but you might have all these different signals there, chemical signals, but the receptor may only fit a certain kind of signal, chemical signal. So in any receptor issue, remember that you have the binding of something with the receptor that fit like two pieces of the puzzle. The cell body, the biggest part of the neuron in terms of size, not length, is the cell body. It receives that information if the receptor is correct. It's sent via the axon, that long skinny part. Thanks, Emma. My non-negative number brain wasn't moving in the right direction. The axon also can have, remember I talked about in another lecture, it can have cushion around it or insulation, and that's called the myelin. The myelin breaks down as we get older. Breakdown of myelin can be accelerated by different diseases like ALS or potentially MS. But it definitely, you get less of it with age. It would be an interesting study to see if COVID had any effect on myelin. I know a lot more people are there's a lot of reasons we might be a little off since COVID. It'd be interesting to study if there was any effect there. The synaptic terminals, synaptic meaning that area between a neuron and another cell. Terminal ending, it's the junction between two cells. A signal is released from the neuron, again, only if that action potential has been sent. And then you have a muscle, a gland, another group of cells there, that they're going to have receptors for the signal that's been released by this neuron. So what we're essentially talking about is we're setting up those metabolic pathways that lead to feedback pathways. So neurons are definitely involved in those pathways. All right, so how is an action potential produced? So now I'll do this the correct way. So as I mentioned, positive and negatives. They sit, those, whatever's released sits in that synapse, or that area between cells. So we know that when we're talking about these signals, we're talking about these signals, we're talking about ions, right? Positive and negative ions, or positively and negatively charged larger molecules or compounds, that these also, these positives and negatives have an electrical output. So remember when you did your ECGs, you hooked up one of your classmates, or perhaps it was you, and you had those little like lines, they were conducting the signals in your body, the positive and negative signals, to produce an ECG printout. We can't feel that amongst each other, right? We can't feel the electricity our bodies produce. I mentioned sharks raise skates. They can feel that. They have a sense where they can feel those positive and those negatives. If you've ever done like a potato, like you hooked up like a potato and to some electrodes to a light bulb and the light bulb went on, all living things have these ions. So all living things have the potential for energy output.
So you could plug in a worm?
Yeah, I mean, it probably would hurt the worm, but I mean, who knows what that potato was feeling, or your banana there, right? You could make a banana fuel something. That's why biofuels really have a lot of potential for energy, because we can produce them over and over and over again, and sometimes quickly if we're talking about like little algae or grass. So an action potential, thank you, Emma, becomes more positive. Well, I was going more positive, but I'm not even going to define myself. I suck sometimes. So let's say, let's do -40. I was going in the wrong I was going in a negative direction. Okay, so maybe you have -39. That won't do anything. Maybe you got a negative, oh yeah, that would, oh my gosh. I gotta think first. -20. Okay, and you have -44. All right, so when we're talking about threshold, if in this case, if our threshold in our cell, we have to add up all of the ions, the K pluses, the Na pluses, the H pluses, the Cl minuses, all of the ions. We add them up. And if they hit -40, it's going to send out a signal. So here, -39 exists right here. So this is going to send out a signal. So that one will send out a signal. I shouldn't put a sign, I should say send. -20. We're moving towards 0. And then we're going, +10. So when we're talking about moving in a positive direction, is that you have from a negative number, your negative numbers move towards 0. And beyond 0, then you go to positive numbers. So if our negative number, like -39, is actually more positive than -40, because it's moving towards 0. So this will send a signal. How about -20? Will that send a signal?
Yeah.
Does it hit threshold? Yep, it goes beyond threshold. +110. Is it going to hit the threshold and move beyond? Okay, good. -44. Does it hit the threshold? No, -44 is up here, right? -44. A -20 is way up here. Does it send the does it hit the threshold? No. Okay, so hopefully that makes up for my mistake earlier. That it doesn't matter. As long as you hit the threshold, you send the signal. The signal for all of these is going to be the same signal. They're just going to hit it and it's going to send a signal. Yeah.
Up to what point does it stop getting negative?
It depends on what's happening in synapse. So it'll like calculate what's happening now. And so let's say that something's released in the environment around the cell, and then it stimulates the positive and negatives to be taken in. And either something happens or it doesn't. Then you might have another cell over here. So let's say this cell is going to stimulate this cell over here. So this cell over here to our cell, it's going to cause this. And it sends out a signal. Now you have another cell over here. And this other cell over here causes the cell body of our cell to become -220, so that nothing happens with our cell. So because you have so many dendrites from a neuron, it is constantly recalculating all of this because it's interacting with a lot of different cells. Does that make sense?
No.
Okay, it's okay. can say no. Okay, so let's go. Okay, so you can see here, here's our cell. It's interacting with this dark orange cell, and it's interacting with this blue cell. So let's say that okay, so let's say our threshold for our light orange cell, this one here, is 0. So we gotta add up our positive and negative ions. If they come to 0, nothing happens. Let's say that when it interacts with that dark orange cell, that the addition of everything in the cell body adds up to -10, which means that no action potential is produced. But now, it's also, like maybe a few seconds later, going to interact with that blue. In the interaction with the blue, so now with the interaction with the blue, our cell body, it adds up to +48. Our threshold is 0. So this is going to send an action potential on. Our cell will be interacting with something at different dendrites. So it's not just interacting with like that dark orange, it might be interacting with that blue, and there's another one here, a purple, and there's a red. And so that every time one of those releases something at their synaptic terminals, at the synaptic terminals there and there, those ions that are released are going to stimulate something to happen here in terms of the addition of the ions, positive or negative. You get that now? Are you sure? Okay, maybe it needs to be absorbed a little, yeah.
So what we need to know is like if it's like greater than the threshold, it sends a signal. Then if it's not, it doesn't send a signal.
Exactly, yeah. So it's just when the body of the cell, when you add up all of the ions, okay, so I'm just giving an example of these. When you add, let's say you have 17, so when you add all of these ions up in a cell body, if they hit the threshold, whatever the threshold is for that cell, if they hit it, it sends an action potential on. The interaction with different cells will cause these numbers to change, depending on what's released into that synapse area. These numbers are constantly changing. Stuff is coming in and out, diffusing in and out of the cells. It's super complicated. So the main take home of this is that when you add up the ions in here, if they are equal to the threshold or more, it sends out a signal. If they are less, when you add them up, if they're less than the threshold, nothing happens. Does that make sense?
Yeah.
Okay. I hope I hope so. Okay. We're going to finish up this in lab, and then we'll have time to study. All right, so thank you for pick up where we left off talking about action potential. I know, you're all in lab mode, I'm sorry. All right, so we left off talking about that action potential means that the cell body moves in a more positive direction. I know this is a little confusing, that we could be talking about negative numbers, but remember, the bigger a negative number is, the less positive it is. The smaller a negative number is, the more it's moving toward 0 or a positive direction. So we have to add all of that up. Threshold is when an action potential is triggered. Oh, threshold for different cells can be different. All of your cells do not have the same threshold. For the exact same cell in each of us individually, we likely have different thresholds as well, because genetically we are different. So for example, people who are autistic or have ADHD might have different thresholds for hearing. They might be more sensitive to sounds, right? So their thresholds will be much lower than someone who could have just like blaring music and be like, no big deal. Whereas like a person with ADHD might be like, ooh, what? Right? They might be really distracted by a large sound. Or hear that sound much like more painful than another person who might hear large sounds and be like, oh, this is great, I enjoy this. So their thresholds are definitely different. So this is very complicated. It's what we call all or none conceptually. It means that when threshold is hit, it sends out an action potential. Action potentials for each cell are not strong or weak. They are exactly the same. So you might be thinking, but then how is it that one person might hear a sound that is, and it causes pain, whereas another person it might cause them pleasure? It means that you have a lot of neurons that are reacting. So you have one that's sending an action potential, right? So you hear a large sound, like a really strong sound. And for, let's say, I know I'm very sensitive to loud sounds. That I might hear that, and what it's going to do is it's going to trigger not only one neuron in my ear, but let's say it triggers 1,000 of them. And all 1,000 are sending action potentials. And all of them combined are causing me to be like, ah. Whereas you might hear a loud sound, a very strong sound, and it only triggers, let's say, 3 action potentials or 3 cells out of 1,000. And so you don't have that many that are firing at the same time. And we're going to get to that too in a second. So this is just another way of thinking about the action potentials and the threshold. So here, let's say your threshold for this particular cell is negative 55. Remember that negative 70 is a less positive number than negative 55. Even though negative 70 is a bigger number in terms of, right, 55, 56, 57, you go up to 70, but we're moving in a positive direction. So this is less positive. Negative 220 would be really less positive. Negative 70 is less positive than 55. Then we go to 40, negative 30, negative 10, negative 2, negative 1, 0, and then we go in a positive direction. Are you? Okay, well, after lecture we'll talk more. Because, yeah, I understand why you're confused, but remember that 0 is our ultimate baseline in terms of positive and negative numbers. Negative numbers get less positive as they get bigger. They move away from positive. Positive numbers get more positive as they get bigger. We'll get you there. So if something sends a stimulus, so a stimulus is sent to our neuron, and in the cell body, when it adds up all of those hydrogens, positive hydrogens, negative chlorides. Everything gets added together, and it causes a cell body to be at negative 70, and our threshold is negative 55. What this stimulus is going to do, it's going to cause that cell to do nothing. So you, for example, let's say that loud sounds, you enjoy them. What's going to happen is that you're going to hear a loud sound, and let's say the cell body adds up to negative 70, you're not going to react. So someone starts blaring music, and you're like, doesn't affect you. Perhaps even you're of a positive reaction, right? Me, when I hear a loud sound, my cell body might add up to negative 20, or it might add up to 40. It crosses threshold, and my reaction is like, oh. And I react to that. We'll talk. So again, how do you tell the difference? How is somebody, if every action potential just sends the same signal on, how do we tell the intensity of a stimulus? So there's two kind of main ways that we tell whether we're going to have a signal sent on or not. I mean, I should say a reaction happens or not. So in an individual cell, there is nothing to tell you about the strength of that signal. But when you have a bunch of neurons in one area react, that means that the strength of the signal gets stronger in more neurons that are activated or send on action potentials. All right, so you might still be like, what does that mean? So two things can happen. Is that one, even if you have one neuron for this particular signal, which usually we won't, but let's say one neuron just gets activated over and over and over and over. So let's say that we're sitting in here and we smell a chemical smell. And that chemical smell doesn't go away. So an action potential is sent in terms of our smell, our sense of smell, and then another one is sent because that smell, that chemical smell is still there, and it's sent again, and the chemical smell is still there, so it's sent again. And the repetition of action potentials by the same neuron being sent tells us something's wrong. The other thing that can happen is many neurons are stimulated. So in that case, we might have all of the neurons of your nose, of your smell, are stimulated at the same time, which causes you like, woo, right? If there is a big chemical smell, and you all at the door areas will smell it probably before we do, and all of your sense of smell is activated by this chemical smell that's coming in, you all on both sides of the room might be like, there's something wrong, right? You wouldn't just be like, I'm not gonna tell her. I'm not gonna tell my classmates, we'll just sit here and suffer. You would react pretty strongly if there was a very strong chemical smell. We might eventually get it, but a lot of your neurons are activated at once. So here, if you have a sibling, you've probably had this happen to you, where your sibling will go like this, over and over and over again, right? So them just touching you might only activate, let's say, one neuron. But because they keep touching it over, that one neuron over and over again, it might cause that one neuron to release an action potential. Every touch releases one action potential from the same neuron. You clearly get triggered by that. Either to one, make the choice, I'm going to ignore them, which we usually learn, it's called habituation, that you stop responding to a signal that's not dangerous. Or if that's like the first time or you're just in a bad mood and they keep doing it, you might react by turning around and punching them. And if you punch them in their arm, in the same place that they were just doing they were activating one neuron over and over and over again, causes you to react, but you punch them, and in you punching them in the arm, it activates a lot of neurons at the same time in this area, right? A punch is going to push in all kinds of neurons. And it's going to cause them to go like that, or it's going to cause them to go like that and then turn around and punch you back, and then there's a big fight.
So my question is, so like, say you're getting punched or poked or whatever, how does your body know to send like the potassium and the sodium and all that stuff like in and out like adequately?
That gets into physiology that's super complicated. The what is stimulated, what is received from the cells here, the signal that's released in the synapse area to the dendrites of our neuron, these cells are going to release a chemical that's received by receptors here. What this cell is releasing and received by this cell is different for what this cell is going to pass on to the next one.
Oh, so they release it based on what they feel?
Essentially, yeah.
Okay.
Because different reactions in cells are going to stimulate different chemicals to be released into the synapse areas.
And then basically the neuron picks that up and tells you that there's an action potential or not?
Yes.
Okay, gotcha.
Yeah, so it's different for every cell, and within a cell, it can be different between two cells and what is released. It's complex. It's super, our cells are extremely complicated, which is one of the cool things about them and kind of mind-blowing. All right, so remember the synapse area, that's where the cells communicate. When we're talking specifically about a neuron and its relationship to something else, we call it synapse. Otherwise, when we're talking about two different cells, we were talking about like the extracellular area or the interstitial area. We just give it a very specific name when we're talking about neurons. All right, so yeah, synapse. The chemicals often that are released are called neurotransmitters because we're talking about neurons. But those neurotransmitters can be hormones, for example. But because we're talking about something that is binding to the receptor of a neuron, we call it a neurotransmitter. We are still talking about the property of diffusion from the synaptic terminal released into the synapse area. You have high concentration in the synaptic terminal to low concentration in the synapse area to low concentration in the dendrite. So it's all diffusion based. If already what is being released by this neuron in the synapse area, there's already a lot in there, it could just make it go and suck back up. It could release it and then get sucked back up, which is also very common in what happens in our neurons as well. Okay, so this is just a blown up, I kind of love and dislike this illustration because your synaptic terminals, what it's really just showing you is they have like little bulbs on the end. If we blow up one of these little bulbs at a synaptic terminal, what you're going to see inside are a lot of vesicles, and the vesicles have chemicals inside of them, meaning that they just have like either we're talking about like a hormone or just talking about a specific kind of ion that's released in response to an action potential. So a particular action potential might cause this yellow to be released by vesicles into the synapse area. There's a little space or gap between a synaptic terminal bulb and the receptor on a dendrite. If that binds with this, it's going to go into the cell body, and then it'll be added up and determine whether a signal is sent on. So neurotransmitters can be a lot of these different kinds of natural things produced by our body like acetylcholine helps our muscles to function, dopamine, norepinephrine, serotonin. These all are different endorphins, which when they are released in cycles throughout our day, help us to feel good or normal, meaning like you feel like not necessarily good, not necessarily bad, but you're feeling decent, right? You're not feeling awful. You're not super happy. You're just kind of like chill, maybe we might call it. Dopamine, serotonin, norepinephrine are released in cycles. Melatonin is released in cycles. Melatonin is naturally produced in our body, helps us to be able to go to sleep on a cycle. These endorphins, all these endorphins, we call them opioids. Isn't that a class of drug?
Yeah.
That class of drugs mimics all of these things that are released in our body naturally, and they mess with them. So that when when you have these being released normally, and then you add a drug to up them, you might feel extra excited, energetic, happy. Alcohol makes them go it makes our GABA receptors go down, which makes you feel like, ooh, I feel chill, right? Marijuana might make you feel, depending on the person, might make you feel more relaxed. So they mess with those drugs. Drugs mess with these. They can enhance or bring them down. So when you take drugs, and depending on the class of drug, it may mess with all of those different kind of like neurotransmitters that make us feel normal. What they're messing with is our reward circuitry. Meaning that I take this drug and it makes, like cocaine often makes people feel high. They call it high because you're like high, happy, excited, energetic. Or it could make your reward would be feeling high, or your reward might be like marijuana or alcohol, which make you feel low, meaning like relaxed, chill. Either way, what you're doing is you're intentionally trying to create a specific feeling in taking drugs. So cocaine, for example, as I mentioned, cocaine messes with our neurotransmitters that make us feel energetic, happy. I'm going to say happy. So what happens with cocaine? So in general, what happens with like dopamine, norepinephrine, and serotonin? They cycle throughout the day to make us kind of feel even keel. When you're excited about something, you might pump out a little more of that. It pumps out the neurotransmitters into that synapse area, and the cell that's receiving it has all kinds of receptors for either dopamine or norepinephrine or serotonin. When you take cocaine, what happens is the first time you take cocaine, they always say like, first time you take it, it's the best high you're ever gonna have. Your brain remembers that first time. Because the first time you've got tons of receptors that are going to take that and send on signals to make you, yeah, woo, feel great, whatever it is. Every time you take cocaine, it kills off the receptors, which makes you able to send on less and less signals, which every time you take cocaine, the ability to send on less signals means you can never get back to the first high with cocaine. But your brain is messed up because your brain's going, I'm going to take more. I'm going to take cocaine, but I'm going to take a little more because then I'll get there to that first high. But guess what? You can't. But your brain, even though you might know this biology, that your brain, you might know, my brain knows, I know, I can never get back to that first high. But your brain still goes, try it anyway. Go ahead, try. Do it. Let's try and do it. And then you take cocaine again. And you're like, darn it, that wasn't like the first time. And then you're like, but I know it will never be like the first time, but I'm going to try again. So that maybe willpower overcomes your knowledge. And so, you know, what happens over time is that certain people genetically are predisposed to being addicted to certain drugs. They can't stop chasing that. Their brain won't let them stop chasing that first high. Another person could do it once and be like, that was cool, I don't ever want to do it again. So a lot of it too, in terms of addiction, changes between us. And you could be predisposed to a drug. You could be predisposed to being addicted to a drug that you never do. But you won't know you're an addict until you submit to that environment. So if you are never intending to drink alcohol in your life, you don't know if you're going to be an alcoholic or not. But if you never drink alcohol, you'll never find out. The other thing that happens is that when you have less and less of these receptors available, in the beginning, you're chasing that high. Eventually, what you're chasing is like, over time, you get to be depressed. You get to go in the opposite direction of that high. And over time, then your brain is like, I just want to feel normal, because you can't feel normal. So it's really hard because people go from chasing the high to chasing just feeling normal because they're so depressed. And when an addict stops chasing both of those things, the receptors can come back. Will they ever come back in the same amount as the first time? No. So they will never get back to that first high. But let's say there's someone who, they're addicted to cocaine, they're rehabilitated, they've been sober for 10 years. One thing could trigger them, and stressful situation, party situation, whatever it is, and they could be like, I'm going to go for that high again. Thank you. Okay, so more and more and more eventually over time is needed to feel okay, unless...
Sorry.
No, it's okay. I'm sorry. The door was locked. And more is just to feel normal. So here, we can look at the difference between someone who, I hate to always say normal, but someone who's, normal means someone who's not using cocaine. So this is what their brain looks like on a daily basis. Cycling of dopamine, norepinephrine, and serotonin. You've got the 3 different colors, right? And so look at the amount of yellow, for example. The amount of yellow, somebody who's been using Coke for 10 days, they're yellow. And the red is gone, right? Do you see any red? So if we're talking about dopamine, norepinephrine, and serotonin being those 3 colors, one of them is completely removed after 10 days. The yellow one, there's not much of it. The blue becomes a lot more. The blue one might cause you to feel more depressed, so you get a lot more of that. Somebody who stops, so this person, they're 100 days past when they used for those 10 days, you can see some recovery back. So these are three different people, just to show you what happens in 3 different people. Okay, I'm gonna move on. Alcohol, it interferes with our, what we call our GABA receptors, our GABA aminobutyric acid. GABA is getting a lot of supplement focus nowadays because it helps you to feel relaxed. Supplements are not regulated very well, so take that all with a grain of salt. You could take a GABA supplement and be like, oh yeah, this totally works on making me feel mellow. It could be totally psychological, like you're like, I think it's happening, therefore it is, when nothing chemically is actually happening.
Is that what ashwagandha is? Like a GABA?
Yes, yes. It works with your GABA neurotransmitter, yeah. So eventually you go in the opposite way of when you're drinking alcohol. Instead of getting like, you want the high, you want the low. And then when you are not getting that like relaxed feeling, the opposite direction is what happens is you get really like nervous and jittery. So it kind of like does the opposite thing that something like a cocaine would do. Alcoholics, what they're chasing is they're just like, they want to stop being so anxious. So they're trying to chase that mellow thing. And so here is the activity of a normal, healthy person. And this is an alcoholic. So again, you can see like those yellows and reds are gone, mostly. The reds definitely. Oh, all of these things, alcohol on young children, marijuana is very similar to this. So I know with marijuana being legal as well as vaping being very big, marijuana use in young people has skyrocketed, and it's causing crazy amounts of anxiety and depression. And some of this is going to be, they believe, irreversible. Alcohol, which is the same as what, well, I should say the same, very similar to what marijuana is going to do. It affects every single area of the brain negatively. So alcohol as well as marijuana is going to be similar in that. COVID and brain shrinkage. They've seen some studies where when people have COVID, the brain has been shrinking. There's a lot more research being done to this. This is very preliminary in what's happening.